Hello, dear students. Myself, Dr. Dhiraj. I do warm welcome to all of you in Maulik online video series. We start with the pharmaceutical branch. In pharmaceutical branch, we take the topic of medicinal chemistry fourth semester, and in fourth semester we take the topic adrenergic drugs. So first of all, we have to understand that what are adrenergic drugs. An adrenergic drugs is related to a chemical compound that especially exerts its physiological actions by stimulation of peripheral sites of sympathetic nervous system. Means these drugs are also called sympathomimetic drugs because they exert their physiological action by stimulation of peripheral sites of sympathetic nervous system. That's why they are also called sympathomimetic agents. And this adrenergic drugs that is derived from the adrenergic nerve fiber. This adrenergic nerve fiber that is basically a neuron for which the neurotransmitters is either adrenaline, that is also called epinephrine, that can be noradrenaline, that is called norepinephrine, or dopamine, or you can say dopamine. So these three types of neurotransmitters, and these three are also hormones, that is the adrenaline, noradrenaline, and dopamine. When we talk in the sense of hormonal, this adrenaline that increases the rate and force of heart contractions. This adrenaline increase in rise of blood pressure. This adrenaline shifts the blood flow to the skeletal muscles. This adrenaline shows mydriasis actions, means dilation of bronchioles and pupils. This adrenaline increases in blood glucose levels. The process is called gluconeogenesis. The process is called gluconeogenesis. Next, we take that what are these adrenaline, this noradrenaline, this dopamine are. What are these chemically? What are these chemically? When we talk about this epinephrine, that is also called adrenaline, norepinephrine, that is also called noradrenaline, that is called the dopamine. These commonly called catecholamines. These commonly called catecholamines. Why they call catecholamines? Because they contain a catechol nucleus that is attached with an amino moiety. That is attached with the amino moiety. So what is catechol? Catechol means any dihydroxybenzene. It can be 1,2-dihydroxybenzene, 3,4-dihydroxybenzene. If in benzene nucleus 2 hydroxy group are present, that is called the catechol, and amino group is attached with this catechol. So this is called the catecholamines. So these catecholamines are epinephrine, norepinephrine, and dopamine because they contain the catechol nucleus and that is associated with an amino moiety. So these catecholamines, that is epinephrine, norepinephrine, and dopamine, these are synthesized from amino acid phenylalanine that is obtained from our diet, that is converted into tyrosine by the enzyme tyrosine hydroxylase. This reaction takes place in cytosol of adrenergic nerve cells. Then the conversion of L-tyrosine to L-dopa is carried out with the help of enzyme tyrosine hydroxylase. This step is considered as rate limiting step, that is also called feedback mechanism, since activity of this enzyme depends upon the amount of stored adrenaline. Then the conversion of L-DOPA to L-dopamine is carried out by the enzyme aromatic L-amino acid decarboxylase. Dopamine is converted into noradrenaline by the enzyme dopamine hydroxylase. Whenever there is an action potential, calcium ion enter into the cell and releases norepinephrine into the synaptic cleft, where it interacts with adrenoceptor and bring about the action. Norepinephrine is converted into epinephrine by the enzyme phenylethanolamine and methyltransferase, where methyl group is added. This step requires methyl donor S-adenosyl methionine. This reaction takes place into the cytoplasm. Whenever there is a depolarization of the membrane, calcium ion enter into the cell called exocytosis of the adrenaline into the synaptic cleft, where it interacts with adrenergic receptor to bring about the action. Now we come on the metabolism, catabolism and degradation part of these catecholamines. Catecholamines are stored in a synaptic vesicles and released into synaptic cleft. Epinephrine and norepinephrine are metabolized by two main enzymes, that is monoamino oxidase MAO and COMT, that is called the catechol orthomethyl transferase. These enzymes are widely present in a system, especially in liver and kidney. MAO present on the outer membrane of the mitochondria and COMT present in the cytoplasm. First step involves oxidative deamination of MAO followed by reduction to form 3,4-dihydroxyphenylethylene glycol, which then catalyzed by COMT to form 3-methoxy-4-hydroxyphenylethylene glycol. So this is the basics of this catecholamines that I told you, that the biosynthesis and its degradation. Now we coming on to this adrenergic receptors, that is also called adrenoreceptors. These are class of G protein coupled receptors, that is the catecholamines like norepinephrine, epinephrine. They are produced in the body, but many medications like the beta blockers, beta 2 agonist and alpha 2 agonist, which are used to treat high blood pressure and asthma, for example, many cells have these receptors, and binding of a catecholamines to the receptor will generally stimulate the synaptic nervous system. Synaptic nervous system responsible for fight or flight response, which is triggered, for example, by exercise or fear causing situations. This response dilates pupils, increases heart rate, mobilizes energy and diverts blood flow from non-essential organs to skeletal muscles. These effects together tend to increase physical performance momentarily. Their action is momentarily. Momentarily, this type of situation arises, that is the flight. Now that we come on the important thing, that is the structure activity relationship of these adrenergic drugs. These adrenergic drugs are categorized into two categories, that is the catecholamines, this is the non-catecholamines. So it is clearly understood from this screen. In catecholamines, there are two hydroxy groups present in the benzene. I already told you, if two hydroxy group are present in benzene, that is called the catechol, and this catechol is attached with the amino moiety. So this is the catechol plus amino moiety. This is called the catecholamines. But in case of non-catecholamines, there are not two hydroxy group. Only there is ethyl amino moiety. So that is called the non-catecholamines. Means which is unable to contain catechol nucleus, which have not catechol nucleus. Now please see the structure. This is the adrenaline, that is the methyl catecholamines, and this is the noradrenaline, that is the simple catecholamines, and this is the ephedrine, that is called the ethyl hydroxybenzene, that is the non-catecholamines. So this adrenaline, noradrenaline, these are the catecholamines. On the other hand, this ephedrine is the non-catecholamines. So now we discuss the SAR points one by one. First point in SAR, that is the sympathomimetic action is shown by both catecholamines and non-catecholamines drugs. Again listen, sympathomimetic action is manifested or is demonstrated by both catecholamines and non-catecholamine analogs. Second point, catecholamine possesses dihydroxybenzene nucleus and non-catecholamines have phenylethylamine ring. Examples for catecholamine, epinephrine, for example, for catecholamines, that is the epinephrine and norepinephrine, and example for non-catecholamines, that is the ephedrine. Next point, for maximum activity, aromatic rings should be separated from amino group by the 2-carbon chain. Next point, increasing or decreasing this carbon chain brings about loss of activity. Next point, substitution on amino group increases beta receptor activity. Primary or secondary amino group have more potent direct acting agonist activity than tertiary or quaternary amines. Next point, substitution on an atom with tertiary butyl group increases beta 2 receptor agonist activity. Example is the clotyrol. Next point, for maximum alpha beta agonist activity, the hydroxyl group must be present on the third and fourth position of the aromatic ring, and also hydroxy group must be present in beta carbon atom. Please see in this figure, that is the 2-hydroxy, and this is the alpha positions. The functional group is NH, and the in the OH it is, that is called the beta position. Now the presence of hydroxyl group on the beta carbon atom makes the compound to have chiral center, which in turn increases the stereoselectivity of the compound, thereby making the compounds better to improve activity. Next point, loss of hydroxyl group in the beta carbon atom results in loss of direct sympathomimetic activity, becomes indirectly acting sympathomimetics. Example is the amphetamine. Next is the removal of hydroxyl group at carbon 4 results in a compound having alpha 1 agonist activity, that is the phenylephrine. Next is the substitution on the nitrogen atom with tertiary butyl group increases beta 2 receptor agonist activity, that is the clotyrol. Next is replacement of catechol nucleus with resorcinol nucleus results in compound having selective beta 2 receptor activity. Example of the drug is terbutaline. Next point, addition of catecholamines and non-catecholamines. Addition of catecholamines and non-catecholamines with other group of compound are found to have alpha receptor agonist activity. Like the aromatic moiety, this is the imidazole ring, this is the brisdunate. So this type of compounds are formed oxymetazoline, clonidine. This is the guanabenzene. Next is the agonist activity is enhanced when the aromatic ring is substituted with halogen substituents like chlorine or small alkyl groups like methyl group, particularly when they are placed in the 2 ortho positions. Now the classification of these adrenergic drugs, these are classified in the following category. First is the directly acting drugs. In this, the different subcategories comes. There are the endogenous catecholamines, that is the adrenaline, noradrenaline, alpha-adrenergic agonist, phenylephrine, methoxamine, nefazoline, xylometazoline, oxymetazoline, clonidine, guanabench, methyl dopa. Next is the dual agonist, oblique antagonist, that is the dobutamine. Next is the beta-adrenergic agonist, isoproterenol, metaproterenol, terbutaline, albuterol, salbuterol, butyroterol, rotodrine. Second is the indirectly acting hydroxyamphetamine, propylhexadrine, mixed acting ephedrine, metaraminol. Next is the endogenous catecholamines are synthesized in the neurons in the chromaffin cells of the adrenal medulla stored in the intracellular vesicles.